much. Certainly, we've had a wide-ranging series of talks, really spanning the gamut of R&D incentives, multiple aspects. So we have a fair amount of time for questions, and so at this point, I'll open it up to the forum members. Jeff.
Thank you all for those great talks. As a public health guy, I'm really intrigued by the idea of using vaccines against antimicrobial resistance. And so I'll direct the question to Drs. Klugeman and McDonald, but would welcome thoughts from all of you. The example of pneumococci is kind of interesting because the vaccine works against the invasive serotypes, and that's largely a function of the capsule. The resistance is based more on a cell wall kind of phenomenon. And so you saw the emergence of some sort of capsular escape variants that also possessed some resistance and didn't retain the full spectrum of resistance that the invasive types that are targeted by the vaccine possessed. I'm wondering if you can just give us some more of your thoughts about the relationship between the mechanism of action of the vaccine and the mechanism of action of resistance and the implications for using vaccines in the future against antimicrobial drug-resistant pathogens.
Sure. So what the data are telling us is that the ecology of resistance is all in the nasopharynx. So the invasive strains, which are really a function of the capsule type, which led them to be included in the vaccine, happen to coincide for the majority of strains, but not all, with strains that were adapted to an ecological niche in the nasopharynx. And the duration of time that these strains spend in the nasopharynx correlates with their exposure to antibiotics and their likelihood of acquiring resistant genes. So in fact, what has happened is that that ecological niche has changed in the nasopharynx, and there are now strains that are less adapted to that locale, but because there's less competition in that site, they're able to colonize, and they're unfortunately able to acquire resistance. It would be a net sum zero game if those strains turn out to be as invasive as the strains that were originally there. Now, so far, this doesn't seem to be the case. But there are populations in which, unfortunately, invasiveness is increased. So the adult in-hospital population of individuals with invasive pneumococcal disease is changing. The fraction, say, who are diabetic has doubled in the last 10 years. So there's an increasing population of individuals who are able to, unfortunately, get invasive disease because of increasing immune issues. So where it'll end, we don't know. At the moment in the US, the net benefit is huge. There's much less antibiotic use, there's much less antibiotic-resistant strains, and the total number of infections is way below baseline. In the UK, for some reason at the moment, they're seeing a massive expansion of these non-vaccine strains which are acquiring resistance, particularly in adults. And it's not really clear exactly why that is happening there, but there's lots of parallels with everything else we're discussing. It's almost like trying to impact the microbiome. And I think what we're learning is that it's extremely complex when your intervention is directed against an ecosystem which has a lot of checks and balances in it.
Okay. Well, I think we'll go around. Jerry, did you have a?
No, it was mostly answered.
Okay. Emily?
Yeah, I just wanted to direct a question to Dr. Barron. At the NIH, we support, you mentioned the very high hurdle to a company getting a diagnostic cleared by the FDA, very expensive and difficult to do the clinical trials. And we have supported at the NIH a couple of specimen repositories for multiple companies to use in order to get their test cleared for an additional indication, gonorrhea and chlamydia from the oropharynx, and the rectum being one that I know of. Would that be useful for diagnostic companies? And could you think of the types of clinical specimens where you'd want to focus your efforts?
It would be extremely helpful to have more access to clinical samples, and we are participating in that trial with the gonorrhea, chlamydia, with a few other manufacturers to get that sample type cleared. But one of the reasons we have problems, at least that I'm familiar with in my company, is that we don't have clinical samples, lots of them available for manufacturing approval testing. So we create matrices that sort of simulate a clinical sample, like a group B strep vaginal rectal swab. We add albumin and God knows what, and we try to make it, but to have real ones would be very helpful. But we need thousands of them. And so these repositories are too limited for us. And we don't have the money really to pay. If you go to a commercial provider, they'll charge $50, $100 per sample or more. So it's just virtually impossible to get access. And so the more access we have to real samples, and you can use seeded samples, of course, but in certain diseases, for example, I would say chlamydia, gonorrhea, if you take vaginal secretion matrix and throw some chlamydia in there, it's not the same thing as a real chlamydia infected vaginal matrix. I hope I partially answered.
Okay, Rima?
Yeah, no, thanks all for great presentations. In thinking about incentives, I think, Dr. Daniel, you're focused on the incentive strategies for new antibiotics, and that's very important. And in following the push-pull and the markets and the product, I wonder how this applies to the range of other incentives for the other approaches that we discussed, whether vaccine, diagnostics, phages, the manipulating the microbiome.
Yeah, that's a great question. We certainly need incentives for the other modalities like vaccines and diagnostics, but most likely won't look the same. So the fundamental challenge with the antibiotics themselves is we want less use of them, right? And we still want the company to be incentivized to develop them, whereas on the diagnostics, we want more use of them. So the sort of the delinkage model may not necessarily work for diagnostics, but we do need incentives for diagnostic use, and I think particularly for vaccines and diagnostics, it's more promising that the healthcare system is moving away from fee-for-service and volume intensity and moving more towards value and outcomes for patients, because in this particular therapeutic area, that's where diagnostics and vaccines can have a fundamental impact on quality for patients and the way that we treat. So there's a way to, and as we move more towards value-based payments, I think the opportunities for diagnostics and demonstrating how diagnostics can result in better care for patients down the road would be an important thing to do. But that by itself isn't going to do it. So we do need much more focus on what the unique economic consequences and issues are for vaccines and diagnostics, and looking in the post-market setting that drives the use of those products. So more activity needs to happen there.
Could I add a follow-on to that? What do you all think about maybe even the potential that I see some of the big pharmas branching beyond just the development of drugs to information technologies or awareness? What might even a model look like managing a population? And I think about this in the microbiome, and tending the microbiome and tending the overall diagnosis and treatment of infections, especially maybe in healthcare, but perhaps also on the farm, through a whole integrated approach where companies would acquire diagnostic tech expertise, information tech expertise, and other proprietary knowledge that could manage the overall health of animals or the health of humans. Just a thought.
Well, I think it is a, it is
I had one model is the IBM, moving from the building of computers to entire service, that model.
Yeah, well, we are seeing I think in the I think Medtronic's a really good example of this in the medical device field, where they're starting to think of themselves as not just a device manufacturer, but a services provider, and they're operating surgical suites in certain hospitals where they're providing end-to-end care for patients. There are concerns about monopolies and what that does in terms of prices, but in this area, the combination of antibiotics, vaccines, and diagnostics does make a lot of sense. There are, I mean, we've tried to get to this in terms of the regulatory approach with companion diagnostics versus not. One of the challenges with companion diagnostics is that then the diagnostic itself is literally tied to that particular drug, and you can't use one without the other. And that may not necessarily be the case. So you need diagnostics that are complementary, but maybe not part of the same regulatory approval process. But I think your question is more towards the company's business model in providing, getting out of just the silo of drug only versus diagnostic only, and sort of doing a more comprehensive approach, which I think makes quite a bit of sense, unless I'm not seeing something. But
I will say that commercial companies are working with drug manufacturers or vaccine manufacturers with companion diagnostics. There's an RSV vaccine being tested with a companion diagnostic to look at the efficacy of the vaccine. I mentioned the Ebola trial. And I think companies are also BioFire FilmArray, for example, and Cepheid both have cloud-based real-time data repository. We've had clinical trials, BARDA-funded clinical trials with Hopkins people looking at flu, real, true reality flu results across the country at various emergency departments to see where is the flu actually happening. So I think that is happening, but I think as Dr. Daniel said, it's limiting, because if you have a co-product, you're limited to use that product in that context, and that doesn't have much financial incentive for you. I'd like to also answer the other question that Rima asked, which has to do with now I forgot what the question was. The incentive for companies. What was it, Rima?
economic incentive.
Economic incentive.
For non-drugs.
Yeah, and I think the model is just, I want to just say something about TB. So there's cost-benefit analyses been published, major articles in CID, about having a rapid TB test and how it pulls negative patients out of airborne isolation faster. And there's plenty of cost benefits, and the FDA even put in the product insert the wording that said 1 or 2 gene expert tests could pull patients out of isolation, could be used. And the National TB Controllers and the American Public Health Labs have published a guideline document on how to take patients out of isolation using a rapid test. And is that test being used in that way? No. Why not? Because all jurisdictions have these very old rules that say 3 negative AFB smears are needed to pull patients out of airborne isolation. And so you are wasting billions of dollars in the United States because of policies have not changed to keep up with technology.
David. Thanks. I had a follow-up question to Jeff's about vaccines and niches and possible future uses. So I want to turn to the gram negatives. And Keith, you mentioned the escape organisms, huge issue, of course, and we would love that this approach might well serve. But of course, as you've said, and Cliff and others have said, we don't know probably enough about some key questions, and those would include, what do those niches look like? What does it take to preempt or displace a resistant organism from one of those niches? And how would you target that subpopulation? And then what happens? Who fills in? And what would that organism be, and what would it look like with respect to resistance? How do you see this playing out?
So I think there are 2 aspects to it. I mean, the low-hanging fruit, obviously, is just to develop a vaccine that protects against invasive disease. And there you don't have to deal with this to the same extent. And so we get enormous pressure to look at both. So clearly, if we could protect neonates or hospitalized patients from invasive Pseudomonas infections, that's fine, but it's only half of the argument. The ultimate would be to interrupt transmission so that you can protect other patients in the hospital. And that, as you say, is a much harder ask. But the notions are complementary. The whole idea of changing the microbiome, I mean, this is one way to do it, just as some of the other ideas around there about using phage and so on, and using probiotics, they're all ultimately going to try and adjust this complex environment. And I think the only way forward is more research. The more we understand the niche, hopefully eventually we'll come up with some low-hanging fruit to alter it.
Kumaran?
Thanks for the excellent presentations, and thanks to the organisers for putting together such a well thought out session. I have a question for Keith and a question for Alan. Keith, at UNICEF, this area of vaccines is close to our heart, but as Sally said yesterday, it's also, we really sort of think of AMR through this lens of the challenges of excess versus access, and she made the point that more people still die from not having access to an antibiotic than they do from resistance. And so I wondered, you sort of present a very interesting view of what's happened in high-income countries mostly when you have increased vaccination in terms of encouraging, but with qualifiers, reduction in resistance. But you presented from South Africa, but what do you think, is there any evidence, or what's the picture in, say, low-income countries where there is much lower sort of use and access to antibiotics? And the follow-up question for that is then, if it's going to be a similar picture, if we can predict a similar picture, are we bringing our efforts to increase vaccine coverage on so-called new vaccines with this concern around AMR? So I'd be interested in your thoughts on that.
Yeah, so there are very optimistic signs that because this particular vaccine interrupts transmission, it's particularly well suited to poor developing countries where you have major problems of access and coverage. So we don't know exactly what percentage coverage you need to interrupt transmission of the vaccine strains, but it's not like measles where it has to be 95%. It's somewhere between 50 and 70% coverage. So the idea is if you can introduce the vaccine, even if the poorest of the poor kids don't get access, they're going to be covered provided sufficient 50% or whatever of that community are covered. So we see prevention as the first and most important thing. So if I get a request from DRC, what are you doing about pneumonia? My first statement is get the vaccine before we try to introduce antibiotics, which are also life-saving. But of course, with antibiotics, you have to get every episode. If a kid has 3 or 4 episodes of life-threatening pneumonia in the first year of life and doesn't get antibiotics for all 3 of them, that's it. Whereas if they get vaccine very early on, it could save their life. But it is, there are 2 sides to this. And so we'd like to push vaccines, and there are better mechanisms to get vaccines into developing countries. Again, this is a whole long debate, but it does turn out that having a vertical system, which is a vaccination program, you can penetrate much more easily than trying to get antibiotics into those countries. But we work on that too, trying to increase amoxicillin exposure, for instance, or access in developing countries is one of our goals in delivery. But we try to prioritize the vaccines over the antibiotics.
Yeah, I'm just wondering, have we also sort of incorporated into our investment case of vaccines this sort of impact on AMR?
That is no, and that was part of my plea for this, for the importance of vaccines. So we are not at the moment, when we look at which vaccines we're going to invest in, at the moment it's all been related to what is the disease burden, what's the mortality that we can prevent, and we have not added an impact on antimicrobial resistance, and we intend to now do that across the board. And I think that will make the value proposition for vaccines even more attractive.
And if I may, I was wondering if I could just ask Alan, we're also, also involved in sort of advanced market commitments, particularly in emergency situations, which has also been triggered by our experience with Ebola. But I wondered, and we have some experience in sort of diagnostics and using advanced market commitments, but in an emergency situation where, yes, there's an immediate need, it needs to be delinked from sort of demand, maybe Gregory also has some views on this. I mean, is that sufficient, or is that fit for purpose, given that, especially say in an outbreak, once that sort of recedes and the diagnostics are used, there isn't really an ongoing market, or do we need sort of other mechanisms in those cases?
I think there are 2 answers to that. One is yes, it's working, and the other is no, it isn't. So for example, there is an emergency use authorization quick route through the FDA for a crisis situation drug or diagnostic to be a very quick path to approval for certain very specified utilizations during that emergency, and it goes away after a certain number of years. But that in itself is not a sufficient incentive for someone to make a diagnostic, because it takes millions of dollars to create a diagnostic. After the EUA disappears, then you still have to do the regular clinical trial. And so, for example, at least my company, when Zika happened, after our Ebola experience, we chose not to even think about making a Zika diagnostic, although we could have, because there just wasn't that long-term return on investment. Now, I just want to make a comment about something you and Keith are talking about. I don't know what developing countries you guys are talking about, but in my developing countries, everybody can get access to antibiotics every day, all the time, down on the street corner. Now, are they any good? Do they really work? I don't know. But we did a thing in Mozambique where we took urine from every incoming patient. We put drops of that urine directly onto a filter paper disc and put it on a petri dish with an E. coli that was susceptible to everything. Every single patient had a big zone of inhibition around their disc because they were all taking antibiotics when they showed up.
Also, just a quick answer to that. Yes, so it varies a lot. Some places like Nigeria and a bunch of other countries, you can get anything you want. The only barrier is, can you pay for it? There are still some parts of the world where you simply can't get anything. But I'd say that the evolution is more towards the direction that you're talking about, where antibiotics will always be available, and it's just a question of who can afford to buy them.
So we're virtually at time, but quickly, Jesse, if you have something you want to.
Yeah, one is to second David's comment about the gram-negatives, and also to say it is complicated to have vaccines against things we're normally colonized with, and there maybe the ambition is to be sufficient to act against, let's say, bacteremia, rather than to affect colonization. Another really interesting observation, I don't know if people are generally aware of it, but there was and I think a tremendous need is in gonorrhea, where again we've run out of drugs, and where there was a meningitis conjugate vaccine that was initiated, I believe, in New Zealand, and with cross-reactivity to Neisseria GC, there was a society-wide reduction in gonorrhea. So I think that's another promising target. And then just couple little tiny comments, if you permit me. I agree that as we transition to real value-based healthcare, it can really help with this tremendously. But one thought I had is, while we're in that transition, which people are trying to prolong forever, as we speak, there are opportunities maybe around pricing, like's happened with oncology. Maybe you pay sufficiently for the drug when it's really needed and it works. So you pay a lot for the drug, but you don't pay for it when it's not needed. Because I think there have to be not just incentives to produce and use these drugs, but disincentives to use them inappropriately. And then finally, on Ellen's comment, as the only, no former FDA or current FDA people, Bernadette's here, but are here to defend the diagnostic process. But I would just say that another side of every emergency I participate in the government or in public health, with diagnostics particularly, is that and even with the current approval process, there are a lot of things that the company thinks work just fine, and then when you get them out in the field, they really don't. So I would just be careful what you wish for in terms of too lax an approval process has public health impacts. And also for industry, it disincentivizes the quality players to make good products. So I think the diagnostic approval process in the U.S. is pretty well balanced.
Just wanted to ask Dr. McDonald if you could quickly tell us a little bit more about your thoughts on phage therapy, because it seems, in what setting you think it would be effective in? Because it seems to me you'd have like the same sort of problems with resistance that you do with antimicrobials.
That's true. It's through alterations in the binding. There is natural resistance. Cocktails would be one. Probably won't be a single phage. Cocktail against a variety of strains. And again, in biofilm applications, it seems most promising, both environmental and on the patient. I will say, and this was already touched on, is that we're going to shift strains always. We have modeling that suggests, and it's not just us, everyone sees this, is that the strains that transmit the best are the ones that tend to pick up the most resistance too. So it is this idea we're targeting that which transmits the best or is most virulent. Not always what's most virulent. I mean, I think that's where we get at this whole colonization, and should we target against colonization? And maybe we could say in general we should target against colonization when we know there's something else less virulent and less resistant that'll take its place. And I think that's been true with strep pneumo. I think we were worried that maybe Staph aureus and even MRSA might occupy that niche in the nasopharynx with the decolonization. It doesn't look like that. It's maybe these less virulent strains. In C. difficile, for example, that right now candidate is against the toxins. Well, that provides opportunity for non-toxigenic C. diff to colonize, and there are those. In fact, that's also been used. So I think maybe in the future, vaccines and the microbiome will really be working in tandem, and all these tools will be applied. And phage would be that way too, I think, if you knock out the most virulent and most resistant. And then there's also the potential, which we can't really touch on, is the ability to manipulate phage with the CRISPR-Cas9 system. And there's some exciting work there where you can actually make the antibiotic resistant ecologically disadvantaged. Happy to talk to you personally about that.
Okay, it's a great session. Lonnie, did you have any comments you wanted to make?
No, I just really appreciate these presentations, and give these folks a hand again. And so we'll reconvene at 10:50.